With its early onset and autosomal dominant inheritance, Huntington's disease dominates the lives of some of the families in New Zealand. Huntington's disease is an example of a trinucleotide repeat disorder. For example, in people with Huntington's disease, when there are more than 35 CAG repeats on the gene encoding the protein HTT, the person will get the clinical symptoms of Huntington's disease. Trinucleotide repeat disorders generally show genetic anticipation, which means their severity increases with each successive generation that inherits them. There are 9 neurologic disorders known to be caused by the increased number of CAG repeats, similar to Huntington's disease. These disorders are all characterized by an autosomal dominant mode of inheritance, a midlife onset, a progressive course, and a correlation of the number of CAG repeats with the severity of the disease and the age at onset. Why do CAG repeats cause neurons to behave abnormally? When the CAG repeats are translated into a series of uninterrupted glutamine residues, which CAG codes for, they form what's called a polyglutamine tract. Such glutamine tracts may be subject to increased aggregation, thus will cause the neurons in which they accumulate to degenerate and die. Listed here are the diseases caused by trinucleotide repeats. Some of them may be familiar to you, particularly Fragile X syndrome, myotonic dystrophy, or Friedreich's ataxia. A specific gene is affected in each case, as many genes contain areas of trinucleotide repeats. Huntington's disease is an autosomal dominant disease, affecting approximately 4 per 100,000 people in our population. The mean age of onset is 40 or earlier, with longer repeats. The predominant symptoms are a chorea, where there are involuntary and uncontrollable jerky movements of the extremities and facial muscles, and cognitive impairment progressing to dementia. In this YouTube video, a doctor interviews a patient with Huntington's disease, and you can see the typical chorea. The abnormal protein in Huntington's disease is called Huntingtin. The expansion of a CAG triplet within Huntingtin results in a different form of the protein, which aggregates abnormally and gradually damages the cells in the brain through mechanisms which aren't fully understood. Huntington's disease primarily damages the brain in 2 areas. The first is the striatum, which is located deep within the brain and is involved in motor control. The outer cortical regions, which are involved in cognitive function and thought processing, also show damage from Huntington's disease, but less severely than in the striatum. These pictures illustrate where exactly in the brain the striatum is. And in this macroscopic picture of a brain, you can see a normal brain on the right and a brain with Huntington's disease on the left. Note the atrophy of the gyri and widening of the sulci, but also the marked atrophy of the midbrain, leading to ventricular dilation. This area here, which is abnormal, is the striatum.